Non-Mendelian genetics. This is topics 4, 5, and 6 in Unit 5. The notes here start on page 35, and we're going to go to the end of the notebook with this. Some things that we're going to be focusing on is explaining how deviations from Mendel's mode of inheritance, from Mendel's model of the inheritance traits, so figure out how they're different. We're going to talk a little bit about how the same genotype can result in multiple phenotypes under different environmental conditions. And finally, we'll talk about how chromosomal inheritance generates genetic variation in sexual reproduction. So let's get started. Non-Mendelian genetics. So we learned all of the rules. Now we're here to break them. Many traits don't follow the ratios predicted by Mendel's laws, and here's a few reasons for it. The first is things have varying degrees of dominance. Not everything is purple and white or red and white. Some things are pink, right? So there's varying degrees of dominance. Many traits are produced through multiple genes acting together. Where we dig into that, we'll learn about epigenetics. Some traits are determined by genes on the sex chromosomes, so that does not follow Mendelian genetic laws. Some genes are adjacent or close to one another on the same chromosome, and they'll segregate as a unit, right? So they'll kind of go together. And some traits are the result of non-nuclear inheritance. So remember, chloroplast and mitochondria has its own DNA. That DNA has traits on it. That is a non-nuclear inheritance trait. So let's talk about degrees of dominance. So alleles can show varying degrees of dominance. In Mendel's experiments, he works with traits that showed complete dominance. So that means purple is dominant to white, right? Homozygote dominant and heterozygote individuals are phenotypically the same. So it doesn't matter if you are homozygote dominant or you are heterozygote, you look the same. With non-Mendelian genetics, we're talking about incomplete dominance, where neither allele is fully dominant. The F1 generation has a phenotype that's actually a mix of the parental generation instead of looking like the dominant phenotype. An example of this is red flowers crossed with white flowers will produce pink flowers. It's neither red nor is it white, but it's pink. It's incomplete. It is a mix of them from the parental generation. So that's what incomplete dominance is when neither of the alleles are being shown. Instead, we get something totally different. That's a mix of the two. Codominance is another sort of phenotypic expression that we see. This is when two alleles that affect a phenotype are both expressed. So human blood groups are a great example of this. Type A blood is actually made up of A and B antigens, so they're both expressed. Sometimes some genes have multiple alleles. So these are genes that exist in forms with more than two alleles. Human blood group is, again, another great example of that, where we have an immunoglobulin A, immunoglobulin B, and then one here that does not produce any antigens. That would be our O-type blood. So let's practice. A mouse that is homozygote dominant is crossed with a white mouse that's homozygote recessive. The resulting offspring are gray. What's the easiest explanation to explain this phenomenon? Well, that's incomplete dominance. Neither is really being expressed. It's kind of a combination of the two. Cattle can be red, that's homozygote, that's all homozygote dominant, that's all red hair. It could be white, homozygote dominant for white hair, or it could be roan. Roan is sort of that red and white hair together. What's the best explanation for this phenomena? Well, it's codominance, because we see both red and white strands of hair. When they come together, they kind of make this weird roan color. If you're interested, this is sort of what they look like. So you can see there's some red hairs that are in there, there's some white hairs that are in there, and they kind of come together. They're both being expressed. So that would not be incomplete dominance, that is codominance. Boom. Great. A red cow is crossed with a roan cow. What would the phenotypic ratio of the offspring be? So we would cross our RR, that's our dominant, our homozygote dominant red hair, and the RW, which is the roan hair. And they organize exactly the same way, right? So we're going to separate the alleles here. We're going to do our test cross, and we find that 1-to-1 ratio between red and roan. That means that 50% are going to be red, 50% are going to be that weird roan combination color. Okay, so that's a great example of codominance. A woman with blood type A has a child with a man that has blood type B. With this limited information, what are the possible genotypes for the woman? What about for the man? They're not looking for you guys to cross these. They're just asking, what are your genotypes? So the woman can either have an immunoglobulin A, or immunoglobulin A, and then no antigen being present. Man can either be immunoglobulin B, or it could be immunoglobulin B and then no antigen. So some traits have two or more genes that are responsible for determining the phenotype. We call this epistasis or epigenetics. This is when the phenotypic expression of a gene at one locus affects the gene at another locus. So an example of this could be color coats in labs in some mice, right? So one gene codes for a pigment, in this case, a black pigment. Another gene codes for an albino, and we have what's called an agouti, which is a combination. So one gene codes for a pigment, a second gene determines whether or not that pigment will be deposited into the hair. So one gene controls another gene. If you have a gene that is for pigment and coat color, but you do not have the gene that suppresses it, then you will have a pigmented coat color, right? So some genes work together. We call this epistasis. There are also things called polygenic inheritance, right? So we're talking about multiple genes and how multiple genes can affect a single phenotype. Polygenic inheritance is the effect of two or more genes acting on a single phenotype. So an example of this is height, human skin color. There are a number of different genes that act together to give those phenotypic traits, right? So as we can see here, this is sort of our Punnett square. It's massive, it's huge, and the number of genes that goes into affecting that color. So 6, 4, 5, these are the numbers of genes that are interacting in these polygenic inheritant traits. So it's complicated, is I guess what I'm trying to say. Okay, some genes that code for phenotypes can be found on the sex chromosome. So we call those sex-linked genes. Thomas Hunt Morgan experimented with fruit flies and determined that specific genes can be carried on sex chromosomes. We call those sex-linked genes. It is a gene that's located either on the X or the Y chromosome. Y-linked genes are specifically found on the Y chromosome. They are not as numerous. The reason for that is the Y chromosome is significantly smaller than the X chromosome. We just don't have enough genes on there to have them be Y-linked genes. There's very few Y-linked genes, so there's very few disorders that are linked to the Y chromosome. One that we do know of is colorblindness, is linked to the Y chromosome. Remember that males have that Y chromosome, whereas females do not. This is one of the reasons why men see a higher frequency of colorblindness than women. Very interesting stuff. There are quite a few X-linked genes, however, genes that are found on the X chromosome. So let's take a look at how we inherit those X-linked genes. Fathers can pass X-linked alleles to all of their daughters, but they will not pass any to their sons. The reason for that is because if we have a Y chromosome, then it is a boy, it is a son. Mothers can pass X-linked alleles to both daughters and sons because remember, the men have the X and Y. So we generally do the nomenclature for this as normal is dominant and affected is recessive. So if we're talking about an affected father, right, we have an X with a normal and an X with a normal. Then we have one X chromosome that is affected and a Y. So because we have that one affected X chromosome, that gets distributed amongst the female children, but not the male children. If we have an affected mother, so we have a heterozygote affected mother and a normal father, we can see that it can get passed to the sons because that X chromosome with the affected X-linked gene is going to be passed to the son with the Y chromosome from the father. Very interesting stuff here. So if it is X-linked from the mother, we can pass it to both daughters and sons. But if the father has an X-linked trait, they can only pass it to the daughters, not the sons. Okay, so if an X-linked trait is due to a recessive allele, females will only express it if they are homozygous for that trait. That makes sense. We have two X chromosomes. If only one of them is a recessive allele, then it's going to be blocked by the other. Because males only have one X chromosome, they will express that trait if they inherit it from their mother. So the reason for that is because they only have one X chromosome. So they don't need the other one to repress it. We have that recessive trait. It's going to be shown in our sons. These are called hemizygous. Since the term is heterozygous, doesn't really apply here. You're not really heterozygote in this trait, right? Because you only have an X and a Y chromosome. Instead, you are hemizygous. Due to this, males are much more likely to have X-linked disorders. Very cool stuff. Let's talk about some X-linked disorders. Duchenne muscular dystrophy is one. This is the progressive weakening of the muscles. Any sort of muscular dystrophy is just a progressive weakening of the muscles. Hemophilia, this is the inability to properly clot blood. That is an X-linked trait. Color blindness, as I said before, that is hanging out on an X-linked trait, and because males only have the one X chromosome, they express it more frequently than females. Okay, so we have X-linked traits. Let's talk about how to turn these on and off. So females inherit two X chromosomes. Again, that's twice as many as males inherit. During development, most of the X chromosomes in each cell become inactive. The inactive X on each cell of a female condenses into what we call a Barr body, named after the guy who discovered it. So in X inactivation, one X chromosome can affect the other X chromosome. One of them will become inactive. It condenses real tight into what we call a Barr body. And what that does is it helps to regulate gene dosage in females, how much of this gene is being expressed. Remember, we have the two X chromosomes. One of them can remain inactive while we express the other. Okay, genes can also be linked, right? Genetic recombination is what happens when we produce offspring with a new combination of genes from parents. We have a parental type. This is an offspring with the parental phenotype, but we also have recombinants. This is what we call offspring with phenotypes that are different from the parents. So your parent has, one genotype, another parent has another, and we recombine those genes to express phenotypes that are different from the parental generation. Offspring with the parental phenotype, however, and again, with this, we're not necessarily talking humans. Think of this more as like flies. Offspring can be born with all parental phenotypes in lower order organisms like that. Okay, so Mendel also observed this, right? Green wrinkled plant is crossed with a yellow round plant. So we have this here. We have a true breeding homozygote recessive and heterozygote for two traits, and they look like that. And when we cross them, we see that 50% look like the parental phenotypes and 50% are a recombinant phenotype. So you can see down here, one of them is going to look just like this one of them is going to look just like this one, and then two of them are kind of going to be mixed up. So this is what we call our parental phenotypes and our recombinant phenotypes. 50% recombination, however, indicates that genes are unlinked or on different chromosomes. Weird. Linked genes are located near each other or on the same chromosome, and they tend to be inherited together. So this here, this 50% recombination, says that they are unlinked. These two genes are unlinked. We can have a yellow wrinkled pea seed. But linked genes are inherited together. Meiosis and random fertilization generate genetic variation in offspring. This is incredibly important to understand. They do it through independent assortment of chromosomes. So chromosomes kind of go their own way. Crossing over in meiosis 1 leads to genetic variation in offspring. We're mixing up that DNA. And random fertilization. Any sperm can fertilize any egg within a species. So here's kind of how it looks. Linked genes typically show parental phenotypes in offspring at a higher than 50% rate. During crossing over, chromosomes from one parental chromatid and one maternal chromatid exchange corresponding segments. So you can imagine if there is a gene that hangs out right here on this chromosome and another gene right next to it that hangs out right there, it's most likely going to be crossed over together, right? Because they're very close. But these genes, while they cross over together, will assort independently or separately than this gene up here. See how they're further away. We only cross over this one segment down here. So the closer they are, the more chances they are of being linked. Boom, right there. So crossing over helps us to explain why some linked genes become separated during meiosis. The further apart two genes are on the same chromosome, the higher probability that a crossing over event will occur between them, and the higher the recombination frequency. So the less these individuals are going to look like their parents. Now, let's remember here, this crossing over event occurs in every chromosome during meiosis. Every single one. It's not just one or two, it's everyone. Okay? So again, the further apart the genes are on the same chromosome, the higher probability that a crossing over event will occur between them, and the higher the recombination frequency, meaning the less you're going to look like the parental type. So we were able to do this experimentally. We performed we didn't, but this dude performed an experiment that allowed scientists to map their genes and their locations. And he drew what's called a linkage map. This is a genetic map that's based on these frequencies. How frequently are these going to be separated and recombined into different cells? The distance between the genes we call map units, and one map unit is equivalent to a 1% recombination frequency. So any genes that are maybe one map unit apart, that's a 1% chance that those genes are going to be recombined together. It expresses the relative distances among chromosomes. So if you see something that is 50 map units, that is a lot closer together to its corresponding gene than say 100 map units. 50% recombination means that the genes are far enough apart on the same chromosome, or they are on two different chromosomes. So if we see more than 50% of these traits being recombined into something new, we can actually pretty confidently say that these genes are far apart on the same chromosome, or they are on two different chromosomes. They are not linked. Great, here's kind of how it looks. So these numbers, you always do it from a relative point. So we're doing this from long Aristae. Aristae are those like little things that come up on the bugs, on like their bug's face. See little bugs, just like that. It's these things, right? So we have short Aristae over here that is marked at a 0 point because again, we have to start somewhere. The higher the number, the more likely that these two genes are not linked. So when we're going through a crossing over period, it is a lot more likely that these are going to be exchanged together than this one and this one is. Practically speaking, what that means is that we will see more flies, because this is Drosophila flies, more flies that have short Aristae and black bodies than we will see that have short Aristae and brown eyes, right? So these traits that are further away will assort independently from the others, but the closer they are together, the higher chances they are of linking. Okay, let's talk non-nuclear DNA. So you have your 23 chromosome pairs in all of your nucleus. However, you have a separate set of DNA in your mitochondria. If you're a plant, congratulations, you learned how to hear because you're listening to this now. And honestly, congratulations, you learned how to use a computer as a plant. Then you have your non-nuclear DNA in your chloroplast. So some traits are located on this DNA. found in the mitochondria of the chloroplast. Both chloroplasts and mitochondria are randomly assorted to gametes and daughter cells. And so remember, when we're doing meiosis or when we're doing mitosis or any kind of cell division, it's not just the chromosomes that are getting replicated and put into different cells. It is all the other organelles as well. So all the organelles are randomly assorted. In animals, mitochondria are transmitted by the egg only, not the sperm. The sperm just doesn't have enough stuff in it to donate mitochondria, right? So all of your mitochondrial DNA actually comes from your mother. This is how, when we get into unit 8, when we start talking about evolution, this is one of the supporting theories in evolution, is that we can actually trace our matrilineage DNA through our mitochondria back to, quote unquote, like the birth of humanity, right, to one single woman. Very interesting stuff. All mitochondrial DNA is maternally inherited. So you only get your mitochondrial DNA from your mother. In plants, mitochondria and chloroplasts are transmitted in the ovule, not in the pollen. So in this class, we don't really get into structure and function and life cycle of plants, but plants don't have eggs, they have ovules. Instead of sperm, they have pollen, but it works all the same way. Therefore, both mitochondrial and chloroplasts determined traits are maternally inherited. You only get them from the ovule, from the egg, whoever's donating that. Okay, chi-square. So this is all well and good, right? Saying that we'll have a 3-to-1 ratio, or we're going to have a codependence, or whatever, all this stuff, right? But how do we know that this is what real life is? So the chi-square is also called the goodness of fit test. It is a statistical analysis used to compare the actual results, what you observe with your eyes, and what we expect to see. This helps to determine whether the data obtained experimentally proves a good fit to the expected data. Practically speaking, this allows us to say what we observe in this experiment here can be used to describe nature, can be used to describe everything else. It determines if any deviations from the expected results are due to random chance or other circumstances. So if you have a goodness of fit test and it's not fitting, we have to figure out whether those are from random chance, like something randomly happened, or whether like we screwed up. And this is a way that we can do that. It's designed to analyze categorical data, all right? So here's how we do it. Oh, I'm sorry, one more. So we use the equation to test the null hypothesis. The null hypothesis says there is no difference. It's a prediction that the data from the experiment will match the expected results. There is no difference from the data that we expect and the data that we collected from the experiment. Here's the formula. It's freaking crazy. So we have the sum x squared, so chi-square, equals the sum of the observed results, so the data we collected from the experiment, the expected result, which is a statistical analysis, that's like your 1-to-3 ratio, that's your 9-to-3 to 3-to-1, we expect to see those. We're going to square it, and then we're going to divide it by the expected results. It's a long process. So that symbol means sum. Here we go. So here's an example. We're interested in examining the trait that allows people to roll their tongues. Tongue rolling is dominant to non-tongue rolling. If you cannot roll your tongue like into a taco shape, you have a recessive trait. You do not have the dominant trait of tongue rolling. So we survey 100 people, and we find that 90 people were able to roll their tongue, and 10 people could not. We're also going to look at genetic data from the parents of all 100 people, and the parental genotypes for all of 100 people was a heterozygote. The null hypothesis states that any difference between the observed and the expected data is due to chance. There is no difference between the observed and expected, and any difference we do see was done by chance, just random occurrences of the universe, nothing that we did. So this is how we solve it. Step 1, determine what your expected and your observed results are. Your observed values are the actual numbers that you get in your data. This is why data is so important. You observe something, you write the data down. No calculations have to be done for this. The expected values are based on probability. We do need to do calculations for the expected values. In this example, we know that all parents are heterozygote for the trait, so we set up a Punnett square to determine the expected numbers of people who can and cannot roll their tongues. So at this point, you'll be doing R, R, we'll do a test cross, and then capital R, lowercase r, where we see that, here we go, phenotypically, we are going to have 3 individuals that display the dominant trait and 1 that displays the recessive trait. So that's your 3-to-1 ratio. We're going to make a table, step 2. The table we make should always follow this format. So we have our observed data, our expected data. Once we get these 2 rows, we subtract them and we put the answer in here. Once we get this answer, we're going to square it. And then once we get this answer, we'll take it and divide it by our expected results, put the answer there. So for this specific experiment, we are going to fill in the table with the trait that we are looking at. So we have our traits. These are individuals who can roll their tongues, individuals who cannot, and then the total count for each. So we perform calculations correctly. This is what our table looks like. So let's just do this one by one. Actually, I'm going to go back and do this. So our observed data shows that 90 individuals could roll their tongue and 10 cannot. That gives us a total of 100. However, our expected data, because of our 3-to-1 ratio, should say that 75 of our individuals should be able to roll their tongues and 25 shouldn't be able to. Just that's our expected results, right? All parents are heterozygote, gives us a total of 100. Now what we're going to do is we're going to subtract our observed value by our expected value. So 90 minus 75 is 15. Okay. Observed is 10, expected is 25. Do not worry if you get a negative number. So that would be negative 15. And that's a total 0. Then we're going to square it. 15 squared, and then this is why the negative doesn't matter, because this is always going to be a positive number. Once you get those numbers, you're going to divide it from the expected. So let's take a look at this chart all filled out. So we have 90 that can roll their tongue, 10 that cannot. But what we expected with our 3-to-1 ratio is 75 should be able to do it, and 25 shouldn't. We got these totals here. Then what they did was we took the observed and subtracted it from the expected, and we got 15. Same thing down here. We observed 10, but we were expecting 25, negative 15. Total here, 0. Okay, then we square it. So we're going to take 15 and square it. That gives us 225. And we'll take negative 15 and square it. Again, gives us 225. Here's our total. Finally, we're going to take this number and we're going to divide it from our expected result. So 225 divided by 75 is 3. 225 divided by 25, because again, we're taking our value here and we're going to divide it from our expected. That comes out to 9. When we total that up, our x squared value is 12. Do you guys see how we did that? It's sort of filling out each row and then filling in the following row. Going down and then across to total it all up. But what does this mean? What does this mean? What is 12 equals x squared? What does that even mean? Well, we have to determine our degrees of freedom first. Our degree of freedom, do they talk about it? Calculate degree of freedom? Okay, we'll get there. Step 4, grab your chi-square table. Okay, we're going to determine our degrees of freedom for our experiment. With our chi-square, you must determine the probability that the difference between the observed and the expected values occurs simply by chance. You need to compare your calculated value to the appropriate value in the degrees of freedom. So remember, we have this x squared value from over here. Now we take this and we're going to compare it to this table. Here's how we calculate our degrees of freedom. Don't ask me why. I don't know. This is just the way we do it. It is the number of categories minus 1. So in our example, for this population, there are 2 categories: individuals who can roll their tongue and individuals who cannot. So our degrees of freedom is 2 minus 1. Our degree of freedom is 1. So we are looking at this row right here. That is our 1. If we were trying to compare 4 different categories, people who can roll their tongue, people who can't roll their tongue, people who have attached ears, people who don't have attached ears, our degrees of freedom would be 3. 4 categories minus 1. Boom, right there, 1. That's what we're looking at. So how do we solve this? The P over here represents confidence levels, right? How confident are you that this is the data that we're seeing? This is the reason why scientists are never 100% certain on anything. Statistically speaking, we cannot be. So 0.5 means that you're 95% confident that your observed data fits your expected data extremely well. 0.1 means that you're 99% confident that your observed data fits the expected data extremely well. We don't live in this row. It's here, it's provided for you just in case, but scientists really live in this 0.5 p-value. Okay, that means that we're 95% confident that our observed data will fit our expected data, even if the numbers are off by a little bit. Even if we expect 25, but we're seeing 27, that still is going to fit our data pretty well. So as a general rule of thumb, we look at that 0.05 row, unless instructed otherwise. These numbers down here, these are your critical values, right? It's a lot of random numbers, but you are concerned mostly with this row and the column in which your degrees of freedom resides. In this case, we're looking at this here. The rest of it can be ignored, okay? So those are our critical values for each degree of freedom. So how do we interpret these results? If your chi-square analysis is greater, if your chi-square value is greater than the critical value, there is a statistically significant difference between the actual results and the expected results. So we are rejecting the null hypothesis. We are saying there is some difference between what we see and what we expect. There's something going on here. There is a difference. So we reject the null hypothesis. Remember, in science, it's not right or wrong. It's accept or reject. We are rejecting the null hypothesis if your chi-square value is greater than your critical value. If it's less than, then there is not a statistically significant difference between what we actually see and what we expect to see. We accept the null hypothesis by saying that there's no difference between what we see and what we expect to see. This is what we want. So for example, we got our chi-square value of 12. This is for the example of the people who can roll their tongue and the people who cannot. Our p-value is greater than what we see in our degrees of freedom in our chi-square table. Therefore, there is a statistically significant difference between what we observed and what we expected. What does this mean? This means we reject the null hypothesis. There is a difference between this. The differences are not due to random chance. There's another explanation here. If your chi-square value comes out to be greater than the chi-square table value, then there's something else going on. Then we have to dig into this further. We have to control for more variables. We have to ask more people, right? We have to do something different. So let's do a practice problem. In peas, yellow seeds, which are dominant over green seeds, we have a cross between 2 plants. Both were heterozygote for seed color. This is what we observed. We had 4,400 yellow and 1,624 green. Does this data fit predicted phenotypic ratio? Well, we're looking at first, we're going to do a test cross, right? We got A, and then A, because they say both parents are heterozygote, and A. This should give us a 3-to-1 ratio. Total number of plants here, you got to add them up. 4, 2, 10, 6. Okay, and then 75% of this would be, which one's dominant here? Yellow. And 25% of this number should be green. That's what we're expecting. Okay, so we set up our table. We have our phenotype over here. It's green, yellow, and then total. So our observed, what do we observe? 4,400 and 1624. Sorry, I just forgot which one's which. 1624. Okay, then we total them up. My total was 6024. I do not have a calculator on hand, but whatever 25% of this is going to go into here, and 75% is going to go into here. Here's all of it together, right? So our green plants, again, we saw 1624 of them, and our yellow plants, we saw 4,400. Here's our total. Then we're going to take our expected, so that's our total, and 25% of them are going to be green. This is, again, what we expect. It's that 3-to-1 ratio, 3 to 1. So we are expecting to see 1506 green, and we're expected to see 4518 yellow. That's our expected value. We double check it over here, make sure the totals match up. So once we do that, we are going to take our observed value and we're going to subtract our expected value. We have those numbers down here. Again, don't worry if it's a negative because we're going to square it. When we square these values, we come out to 13,924 on both ends. And when we divide them from our expected value, we get these numbers here. Adding these numbers up, we see that our chi-square value is 12.32. So taking a look, we're looking at this row here because our confidence levels are P equals 0.5, and our degrees of freedom, we're looking right here. So that number here is 384, and you compare it to your x squared, your chi-square value. Boom, right there, just like that. Because our chi-square value is greater than what the chi-square table tells us, the data does not fit the expected phenotype, so we are going to reject the null hypothesis. We are rejecting the idea that there's no difference. These results are not due to chance. Something else is going on here. Either user error, either these genes are linked, either they're not Mendelian, we don't exactly know. We just know that there's a difference between what we're seeing and what we expect to see. Great. Chi-square is wicked annoying. Once you figure out how to do it, you can just use the chart. So, environmental factors on phenotype. So but what I mean by that is that a lot of these College Board questions will give you your chi-square value and then have you say, are we going to accept or reject the null hypothesis? Or is there a observable difference between, or what is the statistical significance of, you know, a chi-square value that looks like this when your degrees of freedom are 1? That sort of thing. That's what I mean by once you learn how to do this, you'll never have to do it again. All right, so sometimes your environment changes. Various elemental factors can influence gene expression. We call this phenotypic plasticity. Individuals with the same genotype exhibit different phenotypes in different environments. Some examples here are temperature can change the coat color in rabbits and in Siamese cats. So increase the temperature, the coat color changes. We call this phenotypic plasticity. Soil pH can affect flower color. If you have more or less acidic environment, it could affect the flower color. UV exposure can increase melanin production in the skin, right? Folks who are lighter in skin tone, when they go outside in the sun, get a tan. They get a little bit darker. This is phenotypic plasticity. Even though we have the same genotype, nothing's changing there, the environment changes, therefore our phenotype changes. Chromosomal inheritance. So we are going to do quite a bit on chromosomal inheritance and genetic disorders. So your genes, again, are condensed into chromosomes, and some of these chromosomes can cause disorders. So some genetic disorders can be linked to affected or mutated alleles or chromosomal changes. So mutated alleles, this is like the allele that codes for a coat color in mice, right? This is the allele that's affected. So Tay-Sachs disease is a good example of this. It is autosomal recessive, meaning it takes place in the body cells, and it is a recessive disorder. You need two alleles to express it. It's a mutated hexagene. That's just the name of the gene. The gene mutated. In the next unit, we're going to go into mutations and substitutions and deletions and all this stuff. The mutated hexagene leads to Tay-Sachs disease. This is when the body fails to produce an enzyme that breaks down a particular type of lipid. It affects the central nervous system. It can result in blindness. Sickle cell anemia is also a great example of mutated alleles. It's an autosomal recessive disease. So it's actually a mutated hemoglobin gene, and it's caused by a single base pair substitution. It's wild, right? So an A is mutated to a T, and all of a sudden you have sickle cell anemia, right? Sickled cells contain abnormal hemoglobin molecules, causing that weird sickle cell shape. Because we have abnormal hemoglobin molecules, the O2 that generally attaches to the hemoglobin cannot get efficiently transferred to body tissues. It's a whole thing. So let's talk about chromosomal changes. There are some things that can happen during meiosis. Non-disjunction is when chromosomes fail to separate properly in meiosis 1 or meiosis 2, and you end up with 3 chromosomes in one, right? Karyotyping can detect this because we can karyotype your genes. By the way, this is a real karyotype. You can see how it's all kind of weird. Karyotyping can detect non-disjunction because we literally just do a chromosome spread and then count how many chromosomes are homologous. An example of this is Down syndrome, and this karyotype is of an individual with Down syndrome. We call it trisomy 21 because you have 3 chromosomes in the 21 instead of having 2, and that's caused because the chromosomes didn't separate. 3 copies of chromosome 21. Okay, so that's a lot of stuff. That's all non-Mendelian genetics. We are going to be doing chi-square analysis. We're going to be talking about chromosomal changes moving forward. But at this point, you are done with Unit 5 notes. Congratulations. I will see you in class. Thank you so much.